Hello everyone, it's David Leroy here, and this slide presentation is the second in a series that we are doing on the topic of experiments. And in these slides we are going to go over the basics and prepare the groundwork for more advanced study in research methods. So to understand the logic and idea behind experiments, you have to have a good understanding of the terms that we use when we're talking about experiments. And an important term to understand is the one independent variable. So when psychologists and other scientists refer to independent variables, it's important that you have a good understanding of what they mean and that you can spot them in studies easily enough. So independent variables are also called factors, right? So sometimes you'll hear a researcher saying, and the independent variable was X, Y, or Z, and then they may also say there was one main factor in this study. So just be aware that sometimes different terms are used interchangeably, and it doesn't mean that the concept has changed. But in general, the independent variable is distinguished because it is something that is under the control of the experimenter, or often under the control of the experimenter, and it's something that ideally the experimenter wants to be able to manipulate. And so identifying independent variables can be very important, and especially if you're going to be writing about them or expressing your ideas to other researchers, you need to make sure that you name your variables carefully. So try and give them meaningful and intuitive names. It's something that I try and do in my writing. I could say the first variable may just be called something quite bland like group or condition, but if I really mean, that in each different condition they were having different alcohol doses, then I might choose a better name for that variable, which would be alcohol dose, and then the reader can more easily follow along about which variable in particular that you're talking about. So IVs also have levels. So aside from having, one carefully named variable, there will also be levels of that independent variable too. So if you think about an example that I was just using, alcohol dose, this might have meaning for someone who is just starting to read about a piece of research. It might be that you're interested in the effects of different alcohol doses on eyewitness memory. But then it might be the case that you still need more information about that. Okay, so they're going to be manipulating alcohol dose. Exactly how are they going to do that? Well, the different levels in the variable could be a control condition that doesn't get any alcohol, another group that gets a low dose, and another group that gets a high dose of alcohol. So the variable name is alcohol dose, and the different levels refer to the different subcategories of that. Now, independent variables are also classified as either within subject, which is sometimes referred to as repeated measures, or between subject. And it's important that you start to get a feel for the difference between within and between subject variables and what the different advantages and disadvantages are to be able to use those different variables in studies that you may well want to conduct yourself. So the general idea is that a between subjects variable is one where there are different participants in each condition. So imagine following along the lines of the study using alcohol dose. Imagine you had three separate groups of people. The first group of people are in a control condition. They're not going to get any alcohol. The next group of people get the low dose. And then you've got a third group that get a high dose. So they're three separate groups of people, which means the comparisons you're going to be making between the effects of alcohol and the different levels of the variable are going to be between separate individuals. Now this contrasts with the design that we refer to as repeated measures, also called within subject designs, and this is where you've got the same participant in each condition. So imagine you wanted to conduct a study of memory, and then you're going to measure how memory changes over time. So you might take one group of people, show them something that you want them to remember, maybe a video clip of something, and then you're going to come back, interview them immediately, You might call them back into the lab a week later, interview them again, and then you might leave them for a year and interview them after a very long delay. Now, these are the same people that are interviewed each time because you want to track their memory performance over time. So that's called a within subject variable. Independent variables are also classified as manipulated versus subject variables, and other people like to refer to them as random versus non-random factors. Now, manipulated variables are ones where the variable is clearly under the control of the experimenter and there is some ability to randomly assign participants to the different conditions. So if you imagine an independent variable of alcohol dose, how do people end up in the control low dose and high dose group? Well, the experimenter, as people volunteer for the study, will have some method of randomly assigning them to one of those conditions. Now, a subject variable is a factor that's not under the control of the experimenter. Now, variables like this, are legitimate variables and sometimes must be used, but it's important that you're aware of the often lack of random assignment. So maybe you want to do a study between attitudes of people who are short versus tall. So the variable would be height. You'd have two levels, short versus tall. Now there's no way that you can randomly assign people to be short or tall. So you get what you're given there. And the problem with fixed variables like that is that the problem with subject variables is that they can lead to other explanations, right? So if you've got short versus tall people, it might be that you think that the variable is clearly height, but height might be a proxy for something else. It might be a confound in there that you're not aware of or that you should be measuring. So if you're going to put people into short versus tall, yeah, on the surface it does look like height would be the variable, but it could also be other things that cause people to be short versus tall, like nutrition, exercise, and so forth, the height of their parents and so forth. So there could be other factors that require measurement. Now, independent variables are important to identify, and it's also important to be aware that most studies have more than one independent variable, right? And that they're combined to make an incredibly large variety of different experiments. And there are some common ones, but generally speaking, you would be combining within subject and between subject variables. You could have a purely within subject design. You could have a purely between subject design. A mixed design where you use combinations of both types of variable. But at the end of the day, the important thing is that you're easily able to spot the variables that come along in the studies that you read. And researchers will have different ways of explaining those to you. So let's have a look at seeing whether we can spot independent variables in these descriptions of basic experiments. So what type of study is this? Researchers designed an experiment looking at the effects of 3 drugs at treating depression, right? They're also interested in looking at the effects of time of suffering from depression, so that would be short versus long term. So one of the independent variables is going to be drug type, and then the different levels of that are going to be the individual names of the 3 drugs. And then that would be a between subjects variable, and it would be random because you would expect the experimenters, as participants volunteer to take part in the study, to be assigned randomly to one of the 3 different drug conditions. Looking at the amount of time that people have been suffering from depression, it might be an important consideration for the study. But that in itself would be a variable that you might want to call time. The different levels could be short versus long term. It would be between subject because people are going to be in either one of those groups, not able to participate in 2. And it would be a fixed factor. So it's not something that is under the control of the experimenter. Once again, it's something that the participants are going to be bringing to the study themselves. So this is a between subject design, because all of the independent variables are going to have different people in different groups. Let's take a look at this next study. Pause the video if you want to study it for a little bit longer. But in this example, researchers designed an experiment where participants are assigned to one of two memory groups. Right? So they're going to either need to remember words, and some of them will be probably presented as words on a screen. Others, they might be listening to the words presented as audio. So they see or hear lists of words and are asked to recall them in three separate memory tests that are 5 minutes apart. So what are the variables? Well, one independent variable is going to be the memory group, and the different levels of that are going to be word versus audio. And because participants are being assigned to one of the two memory groups, it means that this is a between subject design. So they hear the lists of words and they're recalling them, and here in the description it says that they're going to do three memory tests that are separated by 5 minutes. So they're going to recall the words, wait for 5 minutes, recall them again, wait for 5 minutes, and then recall the list once more. So the memory test is going to be, it could be a test number, and the different independent levels of the independent variable could be test 1, test 2, and test 3, time 1, time 2, and time 3. And that would be a within subject variable because it's the same people that are getting repeated memory tests. And is that factor random or fixed? That's a good question. I think that that would be a fixed factor in this case. Right, a little bit more complicated, but still just as important for you to be able to start extracting information about the different types of study designs that you're reading about. In this example, researchers designed an experiment looking at response time to identifying rotated images appearing on a computer screen. 120 images appear on the screen one at a time, and they appear at different angles, either 0 degrees, 60 degrees, 120, 180, 240, and 300 degrees. So there were also three groups of participants, each seeing the images in different colors. So some saw all of the images as blue, some all of them were green, and some all were red. So maybe the researchers had some idea that maybe the different colors might produce different response times. The researchers also looked at handedness of the participants as well. So they're measuring quite a few things here, and this is where, you know, the combination of 3 or 4 independent variables can very quickly increase the complexity of the design of the study. So what are the so what's the dependent variable? In this case, it's going to be response time, right? So that will just be response time in milliseconds. There's another variable here, which is going to be a degree of rotation, and that's going to be a within subject variable. So it might be orientation, might be a good variable name, and the levels might be 0, 60, 120, 180, 240, and 300, right? So that's going to be a variable called orientation, and it's going to have 6 levels. 3 groups of participants seeing images in different colors, most probably a between subject design, because you have different participants each looking at a different stimuli presented in a different color. And handedness of participants, so that might be, the variable name could be handedness, and then maybe you've got 2 levels, left and right, and that would be a fixed factor, and it would be a between subjects factor as well. So here's just some other examples that we can consider, and this is taken from just a couple of psychology reports, and you'll be reading lots of stuff like this at the moment. It's important to keep testing ourselves to make sure that we really understand the designs of the studies that researchers have been conducting. So here in a results section, the researchers are trying to communicate a little bit about the design of the study. So what are they saying? Total items correctly recalled were submitted to a one-way analysis of variance with delay as a between subjects factor. The main effect of delay was not significant. So here you can see that they're really trying to spell this out for the reader, and be very clear about what their variables were. So they're saying that it's a one-way analysis, right, which means that there's going to be one independent variable. That variable was delay, and it's a between subjects factor. So these are all different people that are being tested at different recall delays. And then after clearly explaining what the variable is, they're able to put in here the result of an analysis. Here's another example. See if you can pick out what's going on in this type of study and look at the way that the researcher has tried to explain the design to the reader. So they're saying to examine hypernesia, and then they've put in brackets a definition of what hypernesia is, which is simply an increase in recall over time. A mixed model analysis of variance. So by saying mixed model, that's telling us that there's bound to be a between subject and a within subject factor in the same experimental design. So a mixed model analysis of variance was performed with the number of details reported, correct and errors, and interview 1 or 2 were within subject factors, and the interview instructions, whether people knew that they were going to be interviewed or not another time, was a between subject factor. You can see there in this example the researcher is trying to explain a reasonably complicated experimental design, but in as clear a manner as possible. Now, the temptation, or one of the things I used to think was, well, if we simply add enough factors to an experiment, we'll be able to answer very, very complex problems. The answer to that is, of course, yes. But the more factors that you add into an experimental design, the more complicated the design is going to be, and the more difficult the findings potentially could be to explain. So the idea here is to think about factors as giving us pieces of individual information. So if you have a study that's got factor A, factor B, or factor C, so that's just 3 independent variables, right? Let's not worry about the levels just for now. But we'll be able to analyze each of those variables in a way that will tell us if there's an effect of factor A, B, or C in a particular study. So if factor A was height, we'd be able to tell whether short versus tall people remember more. If factor B was drug dose, we'd be able to tell which drug produced the best memory effects, and so forth. But what is important to be aware of is that factors interact, and this can be something that you're going to have to perhaps come back to and give some more thought, because it's not immediately obvious what interaction effects are, but they are incredibly important to understand, especially if you're going to be a consumer of psychological science. So an interaction between variables occurs when one level, right, of a variable produces different effects when combined with a level of another factor or variable. So that's the definition. Please read over that and try and imagine what it means. But in general, let's just take a study that's got 3 factors, like we were just discussing, factor A, factor B, and factor C. Now, the interactions between those factors are any and all possible combinations of factors. So factor A can interact with factor B, which is what we would call the AB interaction. It could interact with C, AC interaction, B and C can interact, and then you can get what's called a 3-way interaction, which is an ABC interaction. So let's just look at a 2-way interaction, just so you can begin to understand what we're meaning by interaction. So what we're saying is the effect of one variable is different depending on the level of another variable. So let's say this is height, short versus tall. And maybe we're going to be measuring preference to a new type of shampoo. The shampoo could be pineapple and the other level could be marshmallow. So with the short people, you give them both the marshmallow and pineapple, and they give you their ratings, and they're roughly the same. But when you ask tall people, right, they rate the pineapple and marshmallow as being very different in terms of their preference. In fact, they're rating one much higher, right? And so that means that you've got a different story that you can tell depending on the level of the independent variable you're looking at. And that is really the crux of what interactions are. They can be very complex and they can take quite a lot of time to be able to visualize properly, and you will get better at doing this over the course of your studies and research methods. Just a few more additional comments. It's important to be aware of the distinction that is sometimes made between true versus quasi experiments. Now, psychology is littered with quasi experiments. Now, why are they different to true experiments? True experiments have to involve an element of random assignment, right? And if you have random assignment, you are randomly putting different people into the different levels of conditions in a study. You know, if you get an effect, that it can probably be directly attributed to your experimental manipulation. Studies with fixed factors, which we've been talking about, are often considered quasi-experiments because they don't have that advantage of random assignment. And as I was saying before, an example of that would be if you wanted to measure attitudes of people who are short versus tall. There's no element of random assignment, and as I said before, that can lead to other hidden explanations for the pattern of data that you have in your study being able to explain your findings. And again, we've talked about it before, but it's just important to be aware of the advantages of control groups, why they are particularly important, and if used in a clever way can help us understand complex problems quite well. The control group is a very important IV in some studies. It's a very important variable. Because this is the absence of any manipulation, and it can give you an idea about baseline data, right? And so it gives us something to compare whatever it is that we're interested in to, what happens when the manipulation isn't present. So it allows us to, ask the question compared to what, and that's, whenever we're considering psychological findings, it's also a good idea for you to, just in your head saying compared to, and then try and think about, how you're going to place those findings in context. But anyway, let's have a quick look at, the advantage and logic behind a control group. So imagine a study that finds a sample of heroin users, asks them about previous drug taking behavior, and they say that they have all used marijuana. Now, it's probably no surprise to you that some people would look at that situation or that correlation and conclude that there must be some sort of slippery slope, some relationship between heroin and marijuana use. Maybe people start on marijuana and then they slowly work their way up to using heroin. But that's actually a logical fallacy to think like that. That's the wrong way to look at it. To really, make an argument that, marijuana leads to heroin use, you would need a control group of non-heroin users to look at how much marijuana they use as well. Okay, so that's the same logic as saying, let's say you go to a drug clinic and you've got a sample of heroin users and you ask them all, have you drunk tea? And all of them have had a cup of tea before, and then you might think, well, that means that we know that tea might be involved in leading to heroin use. right? But if you have a control condition, you get some non-heroin users. You ask them if they've had a cup of tea before in their life. They all say yes. Well, then your effect drops away. Suddenly you've got a lot more visibility in seeing what's really going on. Um, so it's another wee nuance here. Sometimes different studies produce contradictory results. So if you're reading a body of literature and maybe you start seeing that some people found some effect, and other people have tried the same thing and they've not found an effect. What you should do is make sure you've got a clear understanding of the independent variables and exactly how they were operationalized. Because sometimes different researchers will use the same name or label for an independent variable, but it might be quite different in terms of what exactly it was that was manipulated or measured. So it might be that you're wanting to conduct a study on stress. You need three groups of people: a control group, a medium stress group, and a high stress group. So it might be that a couple of studies have looked at the effects of different levels of stress. They've found contradictory findings. One might show that stress enhances performance. Another study might show that it decreases performance. But you need to look closely at the independent variables and ask how much stress was applied, how were people put into the different stress conditions. Maybe one study people self-categorized themselves as low, medium, and high, and in another study the researchers might have measured cortisol so that they got an actual, a scientifically precise understanding of how much stress hormone is in the body and so forth. So sometimes unpacking the independent variables could be very illuminating when we look at studies. And also just another note on another nuance here, being aware of single blind versus double blind study. To what If you want to study the effects of, a few different types of drug versus no drug, so drug A versus B versus control, um, how are you going to protect yourself against things like demand characteristics and confounds? So it might be that if you've got a single blind study, it might be that you make sure that when the participant arrives that they don't know which drug they're getting, right? So if you told them, well, you're getting the most expensive and new drug that we're trying to test compared to the other drugs that are previously on the market, it might be the participant feels obliged to say good things about it, in which case it could contaminate the findings in your study. So you might want to keep them in the dark so that as you're taking their blood samples and asking them how they're feeling and so forth, they have no idea which drug that they took. But it might also be the case that, just even if the experimenter keeps the participants blind, the experimenter might act in a different way. Maybe they're really hoping that drug A is going to, produce a new breakthrough. And so maybe they're, subconsciously more pleasant or spend a little bit more time and so forth with people that they've given that drug to in the hope that they'll get more favorable findings. So then you might think, well, actually, to protect against things like that, we need a double-blind study. So we're going to make sure that neither the experimenter nor the participant knows who is in which condition. So it's important to, look for things like that in the science that you're consuming yourself, and also to be aware of things like that should you ever be designing your own study. So I've just talked about this and the different blinding conditions are meant to improve your experimental and scientific precision by removing demand characteristics or opportunities for confounds to explain your data. And just lastly, matching. It's something that researchers sometimes employ when designing their studies. This is a way of reducing unexpected confounds when you've got different levels of variables. So imagine that you've got a variable, it could be alcohol dose, you're randomly assigning people to the different conditions. It might be that just by accident of random assignment, you put older people all in the control group, young people in the high dose group, and then maybe only one gender in another group, right? And so, in actual fact, if you did get differences in between the different groups based on the doses of alcohol, the differences could also be based on the completely different nature of the participants in each of the groups as well. So it might be that you go to the trouble of matching your samples. So you are going to randomly assign people, but you're going to have some constraints, which might mean that there's going to be the same number of males and females in each group, the same age range in each of the conditions that you create, and so forth.